questions? Let's go back and talk about MHC a little bit. For one thing, I said you'll have 6 class 1 molecules on your surface and 6 class 2 molecules on your surface. Not those 3 other class 1 molecules that I said to ignore. You will have one HLA, HLA-A, which is the class 1 molecule that you got from the male, 1 from the female. So that the parents provided 1 each, because as you know, they're diploid, they have 2 each, but the sperm and the egg are haploid, so there's only 1 copy in each one. So you get 1 from dad, mom, 1 from dad, the B, you get 1 from each, 1 from each of the C. So you'll have 6, potentially 6 different class 1 molecules on the surface of most of your nucleated cells. Same goes for the class 2 on the antigen presenting cells, 1 from each parent. Of the 3, that gives you 6 different ones. You could have some that are the same, but not very likely. And yes, they can all be on the surface at the same time with different peptides in their group. Because they're different, the different types that we have, they're going to bind different peptides. Therefore, the peptide you find in HLA-A1 is going to be different than the peptides that you're going to find in HLA-A200 or whatnot. So 6 different class 1 on your cell. They can be if they're all at the same time, all presenting different peptides. And the same for the class 2 on at least the antigen presenting cells. I think those are the main things that I wanted to cover. Were there some other things to clarify? Yeah?
This might be in one of the future ones, but so when you're donating like bone marrow or like for that be the match, when they're matching you, do you have to have all those matching exactly with someone, or is it a specific one thing?
That's a great question. The likelihood of having all 12 match is very low. It's possible, and that's why they're trying to do. They try to get so many people to just provide a little DNA. And they'll just give them epithelial cells from your mouth. They'll find out what your haplotype is, and they can find a perfect match somewhere, hopefully. It's going to take looking at hundreds of millions of people to find it. No, when they do a match, you're not going to be perfectly matched. I have a perfect donor. I have an identical twin brother. So I don't care about my kidneys. I'm wearing them out and I'm going to get them. So, but what they do is they find the best match they can, and then they use some immunosuppressive drugs. Some of them are going to respond a little more strongly than others. So I think that we're still learning. The immunologists and the clinicians are still learning the best methods to use, and eventually we'll have this down pretty low. We'll be able to move the grafts around, the transplantations around. But this is exactly why we think of it. Because as we go through this, we're going to go through the maturation process in the thymus. You see that you make T cells, just like you make B cells that respond to self-antigen, a globular self-antigen. You make T cells that react with each one of these with a certain peptide. It's a random process. The V rearranges with the D and the J, and the alpha chain, the V and the J rearrange, and they just so happen to be the perfect fit to recognize albumin, human albumin peptide, the amino acid 7 through 37 that fits on HLA-A1. You're going to make that guy because you make millions of these things in your thymus. You make millions of B cells in your bone marrow every day. And you're going to make some that are self-reactive. So we have to get rid of those bad ones. And we do a fair job with the B cells. What we don't have to do is as stringent a job getting rid of the self-reactive B cells as we do the self-reactive T cells. Self-reactive T cells would just be, they'll make a mess of everything. They will make so much inflammation in the body that you won't be able to survive. So, anything else about that? A lot of polymorphism and the basis for T cell recognition and the basis for transplantation rejection. All right, so here's our 2 loci again. What we're going to start with is the bottom one, the beta chain locus. We're going to rearrange one of the D's to one of the J's. So it very well could be D beta 2 is going to rearrange to J beta 2 number 5. And when that happens, J beta 2, 1 through 4 are going to be looped out. Same RAG thing, same RAG proteins, same recombination signal sequences that we saw. And I don't know if I mentioned this before, but the only cells that RAG genes get turned on in are B cells and T cells, and only during development. Only when the B cells are developing in the bone marrow, and only when T cells are developing in the thymus. So RAG genes that do this one job, they're only turned on for one time. And I was reading how this happened. I mean, you look at this and you go, how did this ever evolve? And it's, I don't know if you've gotten into retrotransposons and how those things work, but they're the first examples of movable DNA, a DNA cutting enzyme that's encoded, and the specific sequences that it recognizes are on the flanking ends of it. And so basically it just copies itself. It cuts itself out, it goes puts itself somewhere, then it gets copied and cuts itself on. And it looks like the RAG genes were part of a retrotransposon millions of years ago that then got converted into immune system use. So T cells evolutionarily came before B cells. B cells are the latest in the whole process. And I didn't mention this, but it's kind of an interesting thing. Not all animals operate the same way. The mouse system operates a lot like the human system, and that's why we use mice to study the immune system. There are other reasons for doing it. For one thing, mice don't cough or vomit or throw up or anything like that, sneeze. So when you give them a treatment, they can't cough it back out, that kind of thing. And we can also make, twins, congenic strains. So if you wanted a particular strain of mouse that had particular genes, and you can go in and you can get them so that they all have the exact same MHC class 1 and MHC class 2. These mice can be bought. I could buy 200 CVAJ female mice that are 6 to 7 weeks old, all at the same time. They cost about $20 each. So NIH money, the grants to study this stuff cost a lot of money. But you can get them in, and it's just like having 200 twins, identical twins, to study that aren't going to throw up your treatment or cough it up or anything like that. So they're a perfect animal for doing experiments. Not twins, but mice. Anyway, you got me sidetracked there, or I got myself sidetracked. Anyway, in the other systems, so mice and humans are a lot alike. But let's say you go into sharks and look at antibody molecules in sharks. How do they make them? They basically have hundreds of separate sets. So the V gene segments are already hit together, the constant region are hooked together, and it's just duplicated one after another, and they're slightly different. So it's a huge waste of DNA, but it's a very simple thing evolutionarily in that you have separate antibody molecules encoded down the line. Let me get this right. Rabbits. How do rabbits do it? They have very few genes. They just mutate the crap out of it. They go through somatic hypermutation like crazy. They'll make so many B cells, and then so much mutation takes place that they get lots of different clones with lots of different specificities. So there's different ways that this goes on and how it works, but in the end, the innate mechanisms evolved first. Natural killer cells were something that came along a little bit later. And then the T cell receptor came along, T cells came along, and then the B cells were something that came as the last evolutionary step in the immune system. Okay, so we're going to look at this, and this is what I want you to really focus on. You can see which gene is it's the alpha chain. All right, so we're going to look at the alpha chain here again. What I want you to notice is, see what's been stuck in between the V gene segment, right up there, and the J gene segment? That's the whole delta chain locus. Remember I told you there are two types of T cell receptors, the alpha betas and the gamma deltas. And so, if you were to start rearranging the alpha chain, you would basically loop out all of the delta chain. Anything you do, any one of the V gene segments with any of the J gene segments hooked together is going to cause the delta chain locus to be removed. And therefore, you can't make a gamma delta T cell once you've done the alpha chain. But this actually works like the V cells in that the beta chain is rearranged first. D to J and V to DJ on the beta chain, and the alpha chain comes a little bit later. So you're still trying to make that gamma delta chain T cell receptor throughout the development of the T cell. The same things that we saw before in that we have a recombination signal sequences flanking. This doesn't show you the heptamer, the spacer, and the nonomer. It just shows you a 23 on one side and a 12 on another. And it allows you to see that a D and a J are going to hook together, and a V and is going to hook with the D. But in the case of, well, in both gamma delta and the alpha beta, so we still have this 12 to 23 rule, but the T cell actually does some goofy things. And we're not going to get into some of the things they do, but there are always exceptions when it comes to T cells. They do some really crazy things. This is kind of showing you how much diversity we get out of it. And I didn't get into numbers with the immunoglobulin, but you can see here, if you look at the heavy chain and the light chains hooked together with the diversity segments, there's one, the third line down says D segments read in 3 frames. Remember, they're not going to be read in 3 frames because it would put us out of frame shift. You can do that in T cells, so that's one of those things that they do different. Joining segments, and then the P nucleotides, N nucleotides, you've got that going on. Gene pairs are there, almost 2 million, and then you have junctional diversity. So the overall total diversity, just from rearranging the DNA segments in a B cell, is estimated at 5 times 10 to the 13 different affinities, different antigen specificities. Ridiculous. It's an amazing thing just from rearranging the DNA. Then you start mutating it and you add more. Or you just increase, you may not be adding more diversity, but you're increasing the affinity of the diversity you already have. Alpha, beta on the other side, you can see some of the things going on there, put the numbers together, and it's even higher. The total diversity is closer to 10 to the 18 different T cell receptor specificities that are put together from just rearranging the genes of the alpha and beta T cell receptor. So a lot going on there, a lot to recognize. Here again is the complete receptor. We have the alpha beta chain or a gamma delta chain. Both of them have to interact with CD3. We have the gamma and the delta, epsilon, and zeta molecules that are involved in the transduction of the signal. They acquire these in the thymus. When the cell's in the thymus, they have to put the alpha and beta chain or the gamma delta, whichever one it is, with these CD3 molecules to go to the surface. You remember when we talked about IgM, talking about egg molecules, they associated with Ig alpha and Ig beta. And these were the signal transduction molecules. CD3 is doing the same thing. It has to associate with the T cell receptor for it to go to the surface and provide a signal that you've made a functional T cell receptor. Circulation. Well, we're going to have these T cell precursors leaving the bone marrow. So now we're going to go through the maturation. I just kind of showed you the mechanics of what the gene loci look like, what's going to happen. Now we're going to go through the maturation process of the T cell once it leaves the bone marrow. So we leave the bone marrow and we go to the thymus. Once we're in the thymus, it goes through the process of acquiring the T cell receptor and goes through some selection processes. Positive selection and negative selection. Positive is to make sure it's got a functional T cell receptor that recognizes your own MHC molecules. And that's back when I was showing you where do you find class 1 and class 2 molecules. I specifically mentioned, see the epithelium of the thymus? Those epithelial cells express class 2 molecules, whereas typical epithelial cells do not. They also express the class 1. Does the T cell receptor recognize the MHC class, these versions that we have here It has to recognize it first. And the antigens that are being presented on those, there's peptides on those MHC molecules on the epithelial cells in the thymus, but it's not foreign antigen, it's self-antigen. It's just whatever peptide gets loaded on. And if you were to test it and the T cell receptor does not respond at all to any of these molecules, then it's not a functional T cell receptor, and that T cell either has to go through an editing process or it gets destroyed, goes through apoptosis. Then it'll go through a process of negative selection if it's too strong. If it's recognizing self-peptides in context of self-MHC, we can't have that, and so we get rid of it. And then it becomes restricted to CD4, CD8, leaves the thymus, and goes to the secondary lymphoid tissues you see here, going to lymph nodes, spleen, gall, all these different things. And it's a naive T cell looking for its specific antigen. General processes. First thing, the T cell, before it's even called a T cell, it's a progenitor. And it has no T cell receptor, it has no CD4, no CD8, no CD3. It migrates to the thymus where some of them are going to proliferate, but most of the cells that go there end up dying. It's not the most efficient process, but most of the cells that go to the thymus to go through this process don't make it. It starts this differentiation process when RAG genes are turned on, and we start rearranging the DJ of the beta chain. And once that's done, then we will rearrange the alpha chain, we will get the accessory molecules, and go through the selection process that I just mentioned, positive selection to make sure that they're functional, and in a negative selection to make sure they're not too functional. They're not recognizing self too well. Let's see what we're going to next. I do have something that I need to write on the board once we get there. All right. Another picture just to kind of show you the four phases. We have the T cell precursor rearranging the receptor genes in the thymus. We go through the second panel is kind of showing you the positive and negative selection processes still in the thymus. The mature T cells leave there and go to the lymphoid tissue, and you can see they're moving into those different things. And that's where they interact with an antigen-presenting cell that has their particular antigen. And most likely it's going to be a dendritic cell. Not very likely to be a B cell or a macrophage in the lymph nodes, but it'll be a dendritic cell. It gets stimulated, it becomes an effector cell, and unlike the B cells I told you, antibody goes to where the action is. The T cell effector cell has to go where the action is. It's a CD8 T cell that goes looking for virus-infected cells. If it's a T helper cell, it goes looking for that inflamed vascular endothelium, and then goes up in there and searches around for a macrophage that's there that would then present the antigen to it, and it would give it some cytokines and have the cell-mediated response that it's typical. This is showing you, just to get you a picture of what's going on inside the thymus. This is mostly what you're seeing in there is the thymic epithelial cells. Lots of surface area. See the little round things? Those are what we would call the thymocytes. They're T cell progenitors, and all those little round things are kind of going through the process at different stages through the thymus of trying to develop a T cell receptor. I mentioned the Georgia syndrome down here. The Georgia syndrome does not have a completely functional thymus, it may be completely gone, and they do not produce good numbers of T cells, and these individuals have difficulty with infectious agents. So just kind of orient you to what this is going to look like. This next is going to be a cartoon on one side and a real drawing on the other side, where it kind of shows you a cross-section through the thymus and the different areas in it. And so the thymocytes, the T cell progenitors, come from the bone marrow and go up and start to proliferate and then start to go through this process. And you have different types of cells as you move down through the thymus. The thymocytes are going to move down the thymus as they go through the different processes. And there's different cell types as it goes down through there that have different functions in the T cell maturation process. Bone marrow, so this is just in a text form, and then I'm going to have a picture of it. This is text form of what's going on. These T cell progenitors go to the thymus, again, the T cell receptor negative, CD3 negative. This is another term that you'll hear from time to time, double negative. That means CD4 negative, CD8 negative. You hear double positive, it means it has both. There are some occasions that you'll see where it has both CD4 and CD8. It's when they're in the thymus going through differentiation. So they start out with neither. They're going to acquire both, and then they're going to become restricted to one of them. So they migrate to the thymus, they proliferate, the thymocytes receive a signal to stop proliferating and start T cell receptor gene rearrangement. That signal comes from the thymic epithelial cells that are in the thymus. And here's the process that you go through. I don't care that you recognize the CD4, CD25, CD44, and CD25. These are simply markers that the scientists use to track the development of the T cell. What we care about is that they come in there and then they start to rearrange. And here's what I want you to be aware of. They start rearranging beta chain, gamma, and delta. So all of them, once they get in there and they start doing this, they will start rearranging the beta chain, but not the alpha chain. So there's no partner for the beta chain. But it will start rearranging the gamma and the delta chain. And it's kind of a race. Can we get a gamma chain rearranged and a delta chain rearranged before we get just a beta chain rearranged? And that's kind of what it's showing you in the top, or in that panel right below. You see the top brown cell, whatever color that is. That's a T cell that has rearranged both gamma and delta. But more likely what's going on is you have something else. You've got three different versions over here to the right. One is beta chain has been rearranged and that the thing beside it there is a pre-T alpha. Remember when I told you that immunoglobulin heavy chain has to acquire a surrogate light chain? The beta chain has to do the same thing. And so this is pre-T alpha is it's a surrogate alpha chain, pre-T alpha, that associates with the beta chain so it can go to the surface. So that's why it says pre-T alpha beta, and then a gamma underneath it, pre-T alpha beta with a delta underneath it, and pre-T alpha beta. And so those correspond to T cells that have rearranged the beta chain, and in one case they also have rearranged the gamma but not the delta. Another case they've rearranged delta but not gamma. And in the third case, neither gamma or delta have been rearranged. And so once we get to the point where we either have both gamma delta on the surface, or we have beta and the pre-T alpha on the surface, we have another stop, a rest in rearranging. RAG genes are turned off, and we go through proliferation. Again, I mentioned that B cells go through this because you've just spent a lot of time trying to get a functional heavy chain. But and so you need to take advantage of it. We went through a lot of work and put resources into making this immunoglobulin heavy chain, so let's stop proliferate. We're going to do the same thing in the T cells. We've got us a functional beta chain. Let's stop, let's proliferate, make clones, the 5 to 6 rounds of division like we saw in the B cells in the bone marrow before we go to the next step. Question?
So is the pre-T alpha, that's the surrogate thing?
Yes.
Okay.
Yeah, sorry. Now that's the pre-T alpha is this name for a surrogate alpha chain that associates with the beta. And again, in both these cases, these two would have to be there with CD3. These two would have to be hooked together with CD3 and go to the surface. If we get a signal through this complex first, then that's a gamma delta T cell. It is committed to become a gamma delta. If we get the beta chain rearranged, we hook it up with this surrogate alpha chain, and CD3 goes to the surface and we get a signal, then we still have a chance. We still have a chance to be a gamma delta or an alpha beta. But once we start rearranging the alpha chain, we've kind of set ourselves in motion because remember the delta loci is right in the middle of the alpha loci. And once we rearrange an alpha, we loop out the delta and we no longer make delta chains. So we go through 5 to 6 rounds of of proliferation, and then we start rag back up and we start rearranging. And so, as we saw, this one over here, this guy over here, he's committed. That one is a gamma delta T cell, and will leave and go look for its antigen. These others start rearranging, and they'll start rearranging alpha, gamma, and delta. So this one is trying to either get alpha to go with beta, or it's trying to get delta to go with gamma. The opposite here is to get gamma to go with delta, and then this one needs both gamma and delta if it's going to become a gamma delta T cell. But these are the possible things to come out of that, and this is what will ultimately become an alpha beta T cell. This one will too, this one will too, these all become alpha beta T cells. These were not able to get the alpha to rearrange, but did get gamma and delta, so you have gamma delta T cells. So multiple stages to develop the T cell receptor that they're going to have. As I mentioned, gamma deltas are not going to be restricted in MHC like the alpha and betas are. They're the more conventional and important T cell are the alpha betas, not the gamma deltas. The gamma deltas, they're more evolutionarily early. They tend to be lower specificity, and they play earlier roles in the immune responses. Okay, what's next? This is just kind of just putting it in text form. We're going to start rearranging beta, gamma, and delta gene loci. It's a race to see who gets them first. If we get beta chain rearranged, we get surface expression of beta chain with the surrogate alpha chain. That's these two with CD3. If that happens, then we stop rearranging beta, we start proliferating, and then we actually, at this point, express both CD4 and CD8. Now we're a double positive T cell, pre-T cell, still trying to become a T cell. And as I mentioned, it's kind of similar to what we saw in heavy chain previously. This is just showing you again that the delta loci is in the middle of the alpha chain loci, so as soon as you start rearranging them, you're going to lose it. Don't worry about, again, the CD25 and the CD44 stuff. It's just showing you, first thing we're going to do is rearrange the D and the J in the beta chain, and alpha is going to stay in germline. But we also have gamma and delta going through the rearrangement. And then we get a full VDJ rearrangement of the beta chain. And you can see to the right side now we have a protein, the beta chain. And it's going to associate with the pre-T alpha, the CD3, and we're actually going to induce CD4 and CD8 on the surface on the right side of that picture. And then finally, we're going to start rearranging alpha. If we're successful in rearranging alpha, you can see the delta loci has been looped out. And it's lost from that allele. And so that allele will not be able to make a gamma delta, but there is another allele now, because you have two of these in the cells. So in the end, what we end up with here is an alpha beta T cell receptor positive T cell. Let's talk about the diversity. Kind of summarize, and this just kind of goes into the delta loci and gets lost. I just mentioned that. I kind of summarized this with the B cells, and I want to do it with the T cells as well. You've got multiple VD and J segments of the beta chain. I don't want you to get hung up on this other thing, but it adds diversity. The bulleted point there that says T cell receptor beta chain B segment may or may not be used to add more diversity and can be read in multiple frames. Don't worry about that. It's just another diversity piece. Just focus on V to J and VD to DJ rearrangement. That right there is a lot of diversity in the beta chain. And then we have the VJ rearrangement in the alpha chain. And there is some junctional diversity, but in the end, the DNA and the rearrangement of the DNA is enough to develop the diversity that we need. In bold at the bottom, and at least in capital letters, no affinity maturation or class switching in T cells. Once you're an alpha beta T cell, you're an alpha beta T cell. Once you're gamma delta, you're a gamma delta T cell. And we don't go through somatic hypermutation of the CDRs once we get out, like we do with the B cells. And the reason why is because T cells play such a critical role in orchestrating the immune response. If we start mutating a T cell receptor, it's quite possible that it might mutate to the point where now it's recognizing the self-antigen in MHC. I mean, it's down the road, but there could possibly be enough mutation to cause that to happen. All right, I want to talk just a little bit about selection, and then I'm going to have to write some things up here for you all to be aware of. See if there's something that's bothering you about this whole process. All right, so thymocytes bearing T cell receptor, CD3, in both CD4 and CD8, being double positive, must receive a survival signal. So that's the first thing they do is they only last for about 3 days, or they have a half-life of 3 days. So positive selection is that first signal that they get to survive. And basically what it is there's those cells in there, epithelial cells, there's dendritic cells, there's all kinds of things in the thymus that have class 1 and class 2 molecules on their surface. They have self-peptide in those molecules, and they are presenting to these T cells. And if a T cell cannot find an MHC class 1 molecule with self-peptide that it can bind to well enough to get a signal, it's not going to survive. So this is a positive selection process. If there's a weak T cell receptor interaction, that sends a survival signal. All it has to do is it has to be tickled enough by the MHC molecule that it's come in contact with to get that survival signal. Then we're going to move on from there. We acquire CD4 and CD8. Let's see, T cell receptor interaction with self-MHC is stabilized by either CD4 or CD8, and I'm going to show you that again here in a second. The CD4 molecule actually stabilizes the interaction, so the T cell receptor is hooked up against the MHC and antigen, and the CD4 molecule from the T cell binds to the kind of to the side. You know, we show cartoons of it, but how it really looks is another story. But the CD4 molecule stabilizes that interaction. It's kind of a co-stimulatory molecule. It doesn't send the signal so much as it does stabilize it. And CD8 stabilizes the class 1 MHC molecule binding to CD8 T cells. At some point during the positive selection process, either the CD4 is lost or the CD8 is lost. And this is where it's a little hairy as to trying to understand what's going on there. So the T cell has bound to an MHC molecule. It has both CD4 and CD8. One of them stabilized the interaction. Now, how do you decide which one to keep? I don't know. So I don't know if it's a random process for CD4, CD8 loss. Some people have said you actually go through a second round of positive selection. You have a 50-50 chance of getting it right. The 50% chance that you got it right, they go through a second round and they get selected again. You have if the other half of the time you get it wrong and you keep CD8 up, but you're recognizing class 2, then those cells will not get a second signal. I don't know, don't care. All we care about is that during positive selection, the T cell gets a signal and it is stabilized by CD4 or CD8, and in the end, the T cells that survive this process are restricted to one of the MHC molecules. If they're restricted, if they recognize class 1, they have CD8 on their surface and not CD4. If they recognize class 2 antigen, they have CD4 on their surface and not CD8. And we're not going to care how that process happens because it's just still kind of muddy when it really comes down to it. And here's your CD4 and CD8 molecules. And that's just, the CD8 is a 2-chain molecule, CD4 is this 1-chain molecule, and we're not going to worry about the structures of them, just what they do, these accessory molecules. And this is just simply showing you both of them and giving you another kind of visualization of the MHC class 1 with peptide, and the T cell is a CD8 positive T cell that is interacting both with the T cell receptor, the MHC, and the CD8. Now, some of these cartoons, just to keep them simple, they're not showing you all the CD3 and all the other signaling molecules. Don't worry about that. They're there. They have to be there, or the T-cell receptor can be there. That just kind of gives you an idea of what it would potentially look like. Before we move to positive selection, I want you to consider something. The thymus is this little bitty organ behind the heart here that over time gets smaller and smaller, or gets more and more full of fat. And it's kind of limited as to what antigens are being produced in there, right? There are certain proteins that are only made in the pancreas. Let's just talk about insulin or something like that. Only made by certain cell types. Or in the brain, there are certain molecules, proteins, that are only made in the brain. So they're tissue-specific proteins. So how do you go about educating a T cell, or going through negative selection, if you have to? How do you go about getting rid of those cells that might have responded to, that might be reactive to a self-antigen that's not being displayed in the thymus? And I think I might be getting ahead on something, because let me see if we have one more positive selection that just kind of shows you this, and then we'll get into that. This is just simply, again, I just wanted you to have another picture showing you if the T cell receptor is recognizing MHC class 2, an antigen, the CD4 will associate with it, and that becomes a CD4 T cell. If it's recognizing class 1, it becomes restricted to class 1 and is the CD8 T cell. Then we go through negative selection, and that's what I was getting at. We have to get rid of the T cells that have T cell receptors with high affinity for the cell peptides. And negative selection is the way we do that in the thymus. Remember, in the bone marrow, I told you we went through negative selection, and I called it a central tolerance. The central tolerance in the T cell development is also this negative selection, but it's done in the thymus. We also have peripheral tolerance outside of that, but this is the most important for our purposes. We have to get rid of the self-reactive T cells. And they are actually feeling around on their environment, the different MHC molecules they come in contact with, to see if they respond very strongly to them. So we go to, we start checking out the thymic epithelial cells, or the antigen presenting cells, whatever's present there, and if they respond too highly, then we're going to go through a deletion. We'll send them into apoptosis and get rid of them. And so the first two, the smaller bullet points, are just kind of showing you, okay, what sends the signal for negative selection? Most of the time, what it is it's a very strong interaction. Okay, you have a T cell receptor that is binding very strongly to a self-peptide and a self-MHC molecule. High affinity, so that's a strong signal. That T cell is very self-reactive. We got to get rid of it. There are some times where the signal might be induced because it's a very prevalent self-molecule. And so you have a lot of the self-peptide and self-MHC clustered together because it's such a high concentration of the self-antigen, and you have a lot of the T cell receptors on that T cell that are coming and binding to the different places. And so you get a stronger signal just because it's a more abundant self-peptide. That could be going on there as well. But in this case, negative selection is going to be specific for the self-antigen, but it is not foreign antigen specific or dependent. We're still in that phase. When I talked about B cells in the bone marrow, I said you have these antigen-independent phases where it's just going through becoming a B cell, and then when you leave the bone marrow, you get into the antigen-dependent phases. These same thing with the T cell. You're in the thymus. I like to call it antigen-independent because I think of the antigen being the foreign antigen, but it really is interacting with self-antigen in the context of MHC. So you have to be kind of careful about what you're saying there. I like to say it's not antigen specific because it's not specific for a foreign antigen, but in the in reality, it is specific for a self-antigen. So back to where I was talking about, there's only so many of these things that are going to be in there. This is showing you one of the thymic dendritic cells that has MHC class 1. You have a T cell that is recognizing very well the self-peptide in the self-MHC, and the CD8 is binding to it. You've got a strong interaction, and that sends get a signal to go into apoptosis. And so then you end up with a T cell that's going to be deleted. Now, this is where I'll get back to what I was talking about. That's looking at the self-peptides that you typically find in the thymus. There are some antigens that are not going to be present in the thymus, and you have to take those into effect, compensating for those as well. And so what happens is there is a molecule called air, and it gets its name from autoimmune regulator. So A and I, and this is autoimmune, and then regulator, the first RE. And what this is it's a transcription factor. And what it is, it goes into, it can be found, it gets turned on in some thymic epithelial cells. And typically, things like genes like insulin or liver-specific proteins or brain-specific proteins are not going to be turned on in thymic epithelial cells. Air allows these genes to get turned on. So you get insulin made, you'll get other tissue-specific antigens turned on, and you have those peptides made in the thymic epithelial cells, presented on the class 1 and class 2 molecules, so that you have a larger selection of self-antigens to be tested for self-reactivity by these T cells. And you get rid of them. You don't want a T cell that recognizes insulin. You don't want a T cell that recognizes serum albumin. But we get rid of those if they're in the population. Let's see. This next one I'm not going to worry about. It has to do with, it's just talking about the different cell types that are involved in the process. What I do want to move on to is this next part here, bringing up the gamma delta. And you see the flashing mouse example part there. That is for a reason. A lot of this stuff comes about from research on mice, and you can't really make the same correlation to what's going on here in human, because we can't go in and sample fetuses all the time like we can in the mouse situation. But what you'll notice here is that the earliest T cells that come out, that become noticeable in the mouse following, this is showing you days of gestation before birth. You can see that the first ones to come out are gamma delta T cells. And I think the first one is gamma 5, and there's gamma 6. So you have gamma delta are being rearranged in the fetus and are going to be the first ones to come out. And you don't see the alpha beta start coming around until a little bit later. And in fact, it's a restricted number of the gamma delta T cells, with the gamma 5 first and the gamma 6 later. And it says waves of different types of T cells coming in before eventually after birth you have the more diverse gamma deltas. And there's probably a reason for that. It has to do with having these cells actually home to certain mucosal tissues and hang out there as a first-line defense to help out the innate immune system. And then we get more diverse types of gamma deltas later and the alpha betas a little bit later. I think the next thing is just to summarize and talk about alloreactivity and summarize this whole thing. So this alloreactivity gets at the concept that my T cells would recognize your MHC and anything on it as foreign. And I would respond to it. So my T cells have been supposedly educated to not to recognize insulin in context of class 2 and therefore won't respond to it. But if we were to take cells from Destiny and it has insulin in context of her MHC class 2 molecules, there are probably T cells running around in my body that would recognize that as foreign and would respond to it. And that's kind of this idea of alloreactivity. So we're allotypes. And because the MHC can hold about 10,000 peptides each, supposedly, that's an estimate, there's a lot of potential T cells that are out there that could recognize anybody else's MHC molecules with self-peptides that would typically be deleted in the thymus. They would recognize them and respond to them. And the number that has been estimated from mixed lymphocyte reactions is 5% of T cells react to non-self MHC molecules. So it doesn't really matter what's in the non-self MHCs, it just looks foreign. And they respond to it, and they have some affinity for it that will make them react. And there's going to be your graft rejection in a transplantation situation, because there's a lot of MHC molecules on those organs that have been transplanted. This just kind of goes into lymphocyte, summarize this info. It'll take a minute to do that. The T cells, T cell progenitors, come from the bone marrow to the thymus. They move in there. First thing they do is they start to proliferate. At this point, they have no T cell receptor, no CD3, no CD4, no CD8. They don't even look like a T cell. Then they get the signal to start the rearrangement process. And remember that they'll start rearranging the D to J of the beta chain and also the gamma and the delta chains at the same time. And they kind of race to see which one's going to get done first. Eventually what's going to come out is either a gamma delta T cell that is not restricted by the conventional MHC molecules, or a alpha beta that is restricted to CD8 or CD4 by their what they respond to in the MHC molecule, class 1 and class 2. And then they go to the primary or the secondary lymphoid tissues and go through the activation process to start making protein. So that's what we're going to talk about next, is what happens to T cells once they leave the thymus and go to the lymph nodes or other lymphoid tissues to get activated in a primary response, and then what they look like once they're activated. Sure.
This might be totally unrelated, but I'm thinking the process of like dogs and pure breeding dogs, would you eliminate some diversity with inbreeding just because you don't, like if you have similar immune systems?
Yes. The
from
answer is
brothers
yes.
and, okay.
And it happens in human populations too.
Yeah.
I mean, you're going to have some sequestered populations where there's a lot of interbreeding because of because they're the small population and they're too far apart from somebody else. And what ends up happening is the gene pool really does get smaller.
Yeah.
And you end up having things like strange cancers. I mean, rare cancers start being less rare. And we've seen that in smaller populations, smaller towns, even in places like Oklahoma. You'll see an uptick in diseases that are usually rare, but when you start looking at it, it's just been because the diversity has become so narrow that you're not having the amount of, like you said, I mean, it's just.
So that random diversifying that happens that makes everything so diverse, that can be suppressed by inbreeding still.
Well,
because I know some dogs turn out normal.
Not so much with the T cell receptor itself. The genes are probably still there, and you still have the possibility to make all of those, 10 to the 18 different kinds of T cells. The problem is that like the MHC that they respond to becomes fewer and fewer. So if you just have these 12 from this person, and they have 12 from another person, and then there's cousins are interbreeding and everything, you really do end up with a restricted MHC, and we see it. It happens. You end up with fewer MHC molecules, and the ability to respond to multiple infectious agents goes down, and the increase in things like autoimmune diseases
Yeah.
is seen.
Okay,
So,
cool.
yeah, it can happen. It's not a comfortable thing to talk about. I'm from a small town. Okay, let's take about 10 minutes and talk about T-cell activation.